We have four major types of lipids in our diets. Let's take a look at properties of each one and how they are digested. The first one, which is the most abundant, are triacylglycerols. Triacylglycerols have a unique property of being very hydrophobic. They have a glycerol backbone of 3 carbons. We have a fatty acid attached to all 3 carbons. And recall these fatty acids, Fa, are long hydrocarbon chains. The fatty acid chains can be of any length. Typically they are on the order of 16 to 22 carbons long. Now, in order to get this structure inside the body, we need to digest it down, much as we've seen with carbohydrates and proteins, into its subsequent components. So what we are going to do is we are going to use an enzyme-mediated reaction. And these are lipases. Lipases are the enzymes that will digest fatty acid structures off of the triacylglycerol. We have 3 types of lipases. Salivary lipase, which is also termed lingual. We have gastric lipase, and our major one is pancreatic lipase. The salivary or lingual lipase contributes very little to lipid digestion. In adults, gastric lipase contributes approximately 10% to our digestion of lipids in our diet. In infants, it's much greater, for the fact that newborns do not have fully active pancreas yet. But they do take in milk, which has triglycerides, and so their gastric lipase and lingual lipase will make greater contributions. So the lipase activity will digest the glycerol, triacylglycerol, in 2 places. It'll digest here and here on the end fatty acids. The result of this activity is to generate 2 fatty acid structures and 1 2-monoglyceride. So we are releasing these 2 fatty acids from the ends by lipase activity, and the middle one stays, and because this is a glycerol backbone, this is number 2 carbon, this is called 2-monoglyceride with 1 fatty acid attached. Now the lipase does not work by itself, because if you recall back in talking about bile, the triglyceride structures are located in these large lipid droplets. Therefore, the lipase has to be able to work in these lipid droplets. The reaction that's occurring here is a hydrolytic reaction. So in this case, we are adding 2 molecules of water. Recall that this lipid droplet is a completely hydrophobic environment and there's no water available. So the question for many years was, how does this lipase actually perform a hydrolytic reaction in a hydrophobic environment? Turns out that there's another protein that is involved in this reaction. And that enzyme, that cofactor, is called colipase. Colipase is an accessory protein, and it's thought to combine with lipase and be able to act on a triglyceride in these lipid droplets. Now, as an important point of regulation, as the pancreas is generating this lipase and secreting it through the ducts, what is preventing this lipase from doing some serious damage to fatty acid structures and triglyceride structures anywhere? Well, the colipase is actually being secreted by a procolipase. The procolipase has to become activated into colipase, and this occurs by trypsin. So we're looking at the similar sequence here as in protease digestion. The trypsin must become activated first from trypsinogen by the enteropeptidase, and then convert procolipase into the active colipase, and now the lipase can actually work to hydrolyze the fat. The other lipid in our diet are cholesterol. Cholesterol, because of its structure, it's very hydrophobic. No processing is needed, and this is taken up by the protein called NPC1L1, which is the Niemann-Pick C1-like 1 protein. This is a cholesterol transporter. The other major lipid is a derivative of cholesterol, and this is called a cholesterol ester. Now, the cholesterol ester has a fatty acid on it. Let me quickly draw the cholesterol structure. Now recall the cholesterol structure on the number 3 position, 3 carbon on the A ring of cholesterol, has a hydroxyl group. And this hydroxyl in a cholesterol ester actually has a fatty acid attached to it. So I'm going to replace this with a fatty acid. So this is a cholesterol ester. We have a cholesterol backbone, and we have a fatty acid structure, which is considered the ester portion because this is an ester linkage between the carbon and oxygen. In order to generate the digestion products, which is one cholesterol molecule and one fatty acid, the enzyme we are going to use is cholesterol esterase. The final dietary lipid that we have are phospholipids. And phospholipids, recall, are 3 carbon glycerol backbone with our fatty acids on 2 of the carbons. And then on the last carbon, we have our phosphate, and we have attached here the head group. And this can be choline, ethanolamine, serine, and a variety of other head groups. Carbohydrates can also be added. So how do we process this in our diet? We have an enzyme, again, secreted by the pancreas, so it's called pancreatic phospholipase A2. A2 gives it a is a giveaway of where this enzyme cleaves. It cleaves right here. And what we are releasing is one fatty acid and one molecule that still maintains the glycerol backbone, still has one fatty acid, and contains the head group. And this is called a lysophospholipid. If the head group is choline, it's called lysolecithin. Now what this actually does is it allows the fatty acid to be taken up, as we'll see in the next video. And the lysophospholipid, notice our principles here, very similar to a bile salt. This is an amphipathic molecule because we have a fatty acid, which is hydrophobic, and we have the head group, which is hydrophilic. By having this amphipathic head group, this lysolecithin, lysophospholipid structure aids in emulsification.